In this lecture, we are going to examine cell membranes. Common types of lipids encountered in cell membranes are phospholipids, glycolipids, and cholesterol. The fundamental building blocks of cell membranes are phospholipids classified as phosphoacylglycerols. They are also called phosphoglycerides. We can identify four components that make up phosphoacylglycerols, and they are fatty acids, two fatty acid residues, then a glycerol. Of course, fatty acids are attached to glycerol as esters. Alternative to glycerol is sphingosine in molecule of sphingomyelin, which we'll be covering soon. Then phosphate group, which is attached to the remaining hydroxyl group of glycerol, also as ester. And finally, an alcohol that is attached to phosphate as ester. So phospholipids are dicarboxylate monophosphate esters of glycerol. They are similar to soaps in that they contain a polar head and non-polar tails. In this case, polar end of the molecule is composed of phosphoric acid group esterified by an alcohol, so parts that are shown here in red and green. Phospholipids derived from glycerol are phosphoglycerides, and in phosphoglycerides, C1 fatty acid, not alcohol, C1 fatty acid that is attached to carbon 1 is esterified by a saturated fatty acid. In this scheme on the left, that fatty acid is shown as light colored. So that oval is filled in with light color. C2 fatty acid attached to carbon 2, is esterified by an unsaturated fatty acid, and that's shown by a darker fill. And then carbon 3 is esterified by phosphoric acid. Then that resulting molecule is actually a molecule of phosphatidate. So this is phosphatidate. So the phosphatidate does not have an alcohol attached to phosphate. Only a small amount of phosphatidate is present in cell membranes, but it is a key intermediate in synthesis of other phosphoglycerides. Common alcohol components of phosphoglycerides are choline, ethynylamine, serine, glycerol, and inositol. The last one is inositol. Based on the nature of the alcohol, phospholipids are classified as alpha-lecithins, cephalins, phosphatidylserines, and phosphatidylиnositols. They are shown on the next slide. Here are common phosphoglycerides. So general formula is again shown in the upper left corner. And depending on the polar group, they are classified as different classes of compounds. So if that polar group contains choline as alcohol, they are phosphatidylcholines or alpha-lecithins. If it contains ethynylamine, then they are phosphatidylethynylamines or cephalins. Inositol, a six-membered ring with 6 hydroxyl groups, one of which is now bonded as ester group to phosphate group. Then there are phosphatidylinositols with serine as alcohol. So hydroxyl group of serine is one that participates in esterification with phosphate. They are phosphatidylserines. And finally, if molecule of glycerol serves as a bridge, as if it bonds two phosphate groups from two different phosphatidates, then resulting relatively large molecule is called cardiolipin. Sphingomyelin contains sphingosine as the alcohol. So sphingosine has two hydroxyl groups, and on carbon 2 there is amino group. Amino group forms amide bond with a fatty acid, and primary alcohol group, one on carbon 1, is bonded to phosphorylcholine. Sphingomyelin is component of myelin sheath that surrounds some nerve cell axons. An axion is surrounded by multiple sheets of myelin, and they play protective, basically insulating role. Glycolipids contain carbohydrate bonded to a lipid. They are derived from sphingosine and contain a monosaccharide bonded to the primary hydroxyl group. In cerebroside, carbohydrate is glucose or galactose. Glycolipids are always oriented so that the carbohydrate residue is on the extracellular side of the membrane. Membrane lipids are amphipathic molecules. Amphipathic means that they have two ends, hydrophilic and hydrophobic. So membrane lipids have polar hydrophilic head and non-polar hydrophobic tail. That's shown here. In aqueous media, salts of fatty acids form micelles. We have seen that before. However, due to steric requirements of the two non-polar chains, two fatty acid chains cannot fit in the interior of micelle, phospholipids and glycolipids form bilayer membranes. Lipid bilayer membranes form spontaneously by self-assembly process. The process is driven by hydrophobic interaction among the non-polar tails. Lipid bilayers are impermeable to polar species such as ions and polar molecules, but water, because of its small size, can pass through it. Small molecules that are soluble in non-polar solvents can also pass through a bilayer membrane. When we consider shape and size of fatty acids, which are shown here in blue, a phospholipid shown in yellow, and triglyceride, we can see or draw conclusion that they will, in aqueous solution, spontaneously form certain structures. So, fatty acids spontaneously form micelles because that way they expose polar heads to aqueous solution to form hydrogen bonding and non-polar tails towards interior to form London interactions. And so that's the most efficient assembly. On the other hand, phospholipids form bilayer membranes simply because they cannot form micelles. So they form the next most stable structure, which is bilayer membrane. And finally, triglycerides have three large hydrophobic tails and are not capable of forming any regular structures in aqueous solutions. In a lipid bilayer, lateral movement of individual phospholipids is a facile process. However, a transverse movement, so-called a flip-flop, would involve a passage of polar head through non-polar chains and cannot happen under ordinary circumstances. So, lateral movement means movement of one phospholipid on the same side of the membrane laterally, so to another position on the same side of the membrane. That's a very easy process. But flip-flop would be transverse movement, movement from one side of the membrane to another side of the membrane. And that does not happen under ordinary circumstances. So that process under ordinary conditions would proceed with half time of about 10 days, which means for practical purposes it doesn't happen. But there are membrane proteins called flipases that are capable of flipping phospholipid from one side of the bilayer to another. Some passive flipases take minutes to accomplish transverse movement or flip of a phospholipid, and ATP-dependent flipases perform this flip much more rapidly. It is sometimes important to flip it because the fatty acids that are attached to particular phospholipid may be needed for biosynthesis inside the cell, and they may be present just on the outside at the moment. Just like phospholipids, membrane proteins can undergo lateral movement, but cannot undergo a flip-flop. Compared to phospholipids, movement of membrane proteins is considerably more restricted. Presence of lipid rafts, which are semi-solid parts of the cell membrane that contain cholesterol, can considerably restrict movement of membrane proteins. Some of membrane proteins can move relatively freely, but still not as freely as phospholipids, while others are almost completely immobile due to their attachment to a cytoskeleton.